Hello, good evening students. I hope all of you are doing great and good also. Yesterday NEET has been completed and the paper of biology was so easy, and you can do very good in yesterday's paper if you are thorough with biology. And I would like to say that NCERT reading is mandatory for NEET cracking. And one more thing I would like to specify is not only reading of NCERT like mugging up, but you have to understand the concept as well, because there were some thinking questions which were given, and comparatively it is easier when compared to other subjects like chemistry or physics. Now, as I am botany faculty, I am going to take today biomolecules topic, and it's a botany topic, but basically it is related to biochemistry. But it is there in the first year, biomolecules. Let us see what are biomolecules. First of all, molecules that are present in the living system. See here, the name suggests that molecules. So whatever the molecules that are present in the live or living organisms. Bio means living organisms or life. So the molecules that are present in the living organisms like plants, animals, okay, all living organisms, all living creatures, they will be called as biomolecules. They can be like organic compounds or inorganic compounds. So these biomolecules, definitely they are present in the living organisms and they will be doing some or the other functions. Definitely each molecule will have its own respective function in the living organism, what they will be doing in the living organisms. Now let us see where these biomolecules are present. Biomolecules are actually present inside the cells. You know, adult human body is made up of 100 trillion cells. So 100 trillion, can you imagine the count of that 100 trillion? So in that 100 trillion cells, we have in the eukaryotes, we have a nucleus. Eukaryotes means those which have the true nucleus. So in that, one nucleus is there, and apart from the nucleus we have cytoplasm as well. So in the cytoplasm or inside the nucleus, these biomolecules are present. Either they can be there in the nucleus of a cell or they can be there in the cytoplasm of a cell. Generally they will be there inside the cells in these two regions. Or else when we have the circulatory system, that blood will be circulating throughout our body. So in the blood also, so many biomolecules will be there, but generally they will be smaller in size. So that is about the biomolecules, where they are located in the organisms, living organisms. Basically inside a cell, they are present inside the nucleus as well as in the cytoplasm. Outside the cell, they will be circulating like through the blood, okay, to different parts of our body. If you take the plant also do have conducting tissues like xylem and phloem, which you are aware because after anatomy. In anatomy you have learned that xylem is the water conducting tissue and phloem is the food conducting tissue. So inside the xylem or inside the phloem, these molecules will be circulating throughout the plant body. So molecules that are present basically in living organisms are called as biomolecules. So various biomolecules are present. As I told you, like organic and inorganic substances also can be there. Let us see different types of biomolecules here. I have given the central one, biomolecules. These are the biomolecules which are categorized into metabolites. Metabolites means which are going to perform certain activities, metabolic activities. So metabolites in the sense which are capable of doing metabolism. Some kind of activity will be happening inside the bodies of the living organisms, which are called as together metabolism. That means at any point of time in your cell or in your body, some molecules will keep on forming from other molecules, or some molecules will be broken down continuously. If they are getting formed, that kind of process is called as anabolism. See some example like A and B. Let us take two molecules are there, A and B. These two molecules are getting fused to form a compound C. So this kind of mechanism where A, B are combined to form C is called as synthesis or anabolism. So in the synthesis, what is going to happen is the two molecules will be combining to form one more compound. That is the synthesis process or anabolism. Next, one more thing, if this C is broken down to form something else, this C will be broken down to release P and Q. Okay, this molecule is breaking down into not into A and B, or you can imagine A and B also, but it is broken down to form P and Q, some other molecules, because that formation is a different step and breaking down is a different step. So if this C is broken down into P and Q, this is called as breaking down or breakdown or catabolism. So the molecules in our body keeps on, any point of time, they keeps on going to be synthesized or they are going to be broken down to do some of the functions in our body. So basically we have only two metabolisms like anabolism and catabolism. This is the first one, anabolism, and this is the second one, catabolism. So in anabolism already synthesis of compounds will be happening, and in catabolism breakdown of compounds will be happening. That molecules or compounds, whatever you take. So molecule means it is formed by two atoms, and compound means which are having several molecules. So if this C molecule is getting formed from A and B, that kind of mechanism is called anabolism or synthesis. If the C is broken down to form P and Q, then that process is called as catabolism. These two are the steps of metabolism. So metabolism includes both anabolism and catabolism. But in a given point of time in a cell, any one will be happening or they will be linked to one another. See, for example, I have linked this anabolism and catabolism. Either they can happen singly or like in continuation in a link they can occur in the living bodies. So continuously inside our cells metabolism will be happening, and in that metabolism whichever the molecules that are being used or participated, they are called as metabolites. So basically metabolites means they are the molecules or substances or compounds which are participating in the metabolism like anabolism and catabolism. This is a continuous process. The living organisms are said to be continuously in the living state because of this metabolism. This is a unique feature of living organisms. All the living organisms, they will be showing metabolism. Whatever the living organism you consider like bacteria, algae, fungi, whatever the living organism like plant, animal, humans, every living organism will be showing the metabolism. This is unique feature of living organisms, which is shown by all living organisms. So the molecules, as I told you, the molecules which are participating in metabolism are called as metabolites. These metabolites, they are being classified into two types. We have two types of mechanisms. So anabolism and catabolism, and these metabolites are two types, secondary metabolites and primary metabolites. The primary metabolites means we have definitely the use of these primary metabolites in the living cells, without which the cell cannot live. So primary means first function. So whichever the metabolites, they will be doing the main functions, they are called as primary metabolites. Okay, whichever the molecules or metabolites that are doing the primary main functions without which the cell cannot survive, the cell cannot do the functions, that kind of important substances or very important substances are called as primary metabolites. They are present in all animal tissues, okay, they are present even in the plants also. It depends what living organism you are considering. So they are mainly present in the living organisms to perform specific functions, and without which the cell cannot live or survive. They are, for example, sugars. Sugars are required for the primary functions of this cell, and then lipids also will come under that category, and amino acids also will come under that category. So primary metabolites means they are present in the cells to perform definitely important functions, and these are the three examples: sugars, amino acids and lipids. Now coming to the secondary metabolites, these are of not that much use for the cell, but simply they are present, and these secondary metabolites we can make use. Like for example, you must be aware of the rubber what you are using, gum, rubber, eraser is made up of rubber, and even gum is there which is extracted from the plants that we will use to stick the papers and all. You are aware, no? So that kind of gums, okay, rubber, okay, some kind of oils what we are using, the plant is simply producing, and the cells can survive without them also. There is no problem for the cell even if we remove that. See, for example, we are extracting the rubber containing latex from the plant, but even then the plant will survive. That is the nature. So here the secondary metabolites are not that important like the primary metabolites. These are doing some specific functions only, but they are not important for the cell. Without them also the cell can survive. Even if we remove all the secondary metabolites also, the cell can survive. There won't be any problem at all. So that kind of substances are called secondary metabolites. They are flavonoids, alkaloids, gums and rubber essential oils. So we have basically the metabolites categorized into two types: primary metabolites and secondary metabolites. Let us see that. Metabolites are of two types: primary metabolites and secondary metabolites. These are definitely compulsory, they are required. Requirement is compulsory. Here, not required compulsorily. Okay, by the organism. Not required compulsorily by the organism. These, they do specific functions. Not known functions. The functions of secondary metabolites in the living organisms is not known or not understood yet. So we don't know exactly what are the functions of secondary metabolites in the organisms. But the man is the cleverest mammal on this earth. So what we are doing is we are extracting the secondary metabolites from the plants, and we are making use of their secondary metabolites for our own use, for the welfare of mankind. We are taking the rubber, oils, everything from the plants, and we are making use of them in our daily life. Okay, that is the use of that secondary metabolites, but not for that particular living organism from which we are extracting those products. That is about primary metabolites and secondary metabolites. Primary metabolites examples, as I have shown you in the before slide: amino acids, okay, then sugars, then lipids. These three will come under the primary metabolites, whereas other things like oils, rubber, gums, essential oils, okay, rubber, gums, etc., they will come under secondary metabolites. So like this, the metabolites are classified: primary and secondary. So that is about the metabolites. You can see here the metabolites are given. So we have completed the metabolites. They are the substances or molecules that are present in the living organisms with the specific functions, without specific functions, without which we cannot survive. Without this we can survive. There is no problem at all for this cell. Now the biomolecules, how we can identify these biomolecules, how we can extract them from the living cells. The basic mechanism is like we have to take a particular leaf tissue, See, all of you can see here one kind of pestle and mortar. The one which is used actually to prepare chutney at your home or something like that. But this is a pestle, the one with which you are grinding, and this basic one, basal one, is a mortar. So by using this, okay, if I give you a plant leaf, for example, I'll be giving you a leaf. So that leaf, you are going to take it in this mortar and you are going to crush it. While crushing, we have to add the trichloroacetic acid. Just take a leaf in this, okay, add trichloroacetic acid. So while adding that trichloroacetic acid, you have to just grind the leaf and you have to get the extract. Some liquidy thing you will be getting, that you have to filter it. So how you are going to do that filtering? See here, in this we have collected that leaf extract or orange juice or any juice, whatever we have prepared. That juice we have to collect it here, and then we have to pour it through the funnel, and which is having a filter paper. Generally filter papers like tissues like that, they will be there, but in the chemistry lab we will be seeing the filter papers. So that filter paper you have to keep it inside this funnel, which will be acting as a filter basically, and through the funnel, if you pour this liquid or leaf extract which we got in this, the liquid portion will be collected here at the bottom, which is soluble in the acid. So here basically we will be getting two parts. In the down, it is called as filtrate, which has been filtered through this funnel, or the filter paper is called as filtrate, that is collected here. And whichever it is present here, this is called as retentate. And this in this flask or the bottom conical flask or a jar, wherever you are collecting this filtrate will be present in this. And retentate will be present in that filter paper only, that paper what we are using or which we are using it as a filter, in that only it will be there. So this which is there in that funnel is called as retentate. The one which is collected in the jar, it is called as filtrate. And I'll tell you one more difference here. The thing is, when your mom is making tea, okay, when you visit a tea shop, the tea filter will be there, and the tea filter will be filtering out the liquid down in the cup. And the ones which are caught in that tea filter, they are nothing but tea granules will be collected. So that tea granules which are collected in the tea filter are nothing but the retentate, and the tea which is collected in the cup, bottom cup, that is the filtrate. You can understand in that way better. So here the same thing, the filtrate will be there and the retentate will be there. In the retentate, the best part is the molecules which cannot be filtered only will be there, no? See, for example, if it is filtered, obviously it will be collected in the down portion filtrate. That means in the retentate, obviously big molecules are present, or larger particles are present. So here always you will be having larger particles. Whereas here in the filtrate, because it is filtered through this, it will be having smaller particles. So if they are very small, which are soluble in the acid, they will be filtered out and they will be present in the filtrate. If they are very large and which cannot be filtered, all of them will be there in the retentate portion. So here retentate means it will be having the larger molecules, and filtrate means it will be having the smaller molecules. And one more term we can write here, that they are like acid soluble pool, because we are using the trichloroacetic acid. This acid what we have used before is a trichloroacetic acid. If you see there, this is the trichloroacetic acid here. So the acid soluble ones will be collected in the filtrate. So we can clarify saying that filtrate will be having acid soluble pool. Whichever the substances that are filtered, okay, they are there called as filtrate and they are soluble in the acid. That is why we can call the filtrate is nothing but acid soluble pool. Then retentate is called as, which is left over in that filter, is called as acid insoluble pool. maybe they are not soluble in the acid, okay, that is one reason, and there are larger particles also which cannot be filtered. So because of that they did not filter into the jar. So because of that we can say retentate is acid insoluble pool, or it will be having the larger particles. Filtrate is the one which is having smaller particles, definitely smaller molecules, and it is having all the substances which are acid soluble. They are soluble in the acid. Which acid is that? Trichloroacetic acid. So take any living tissue, grind it in the mortar and pestle. In this, any living tissue, I have taken the leaf, but you can take any kind of tissue, and just you have to grind it here with the help of trichloroacetic acid, then do the filtration technique to separate the molecules based on the sizes and solubility. The main techniques which are involved in this filtration are the solubility, and size of the particles. Based on 2 points, the filtration technique is there. That is, one, size of the molecules, and the second one, solubility, whether it is soluble in the acid or not. So after this we got 2 portions. One is the filtrate, which is acid soluble pool. Another one is retentate, which is called as acid insoluble pool. Can anyone tell me which one will be having the larger particles, retentate or filtrate? It is the retentate, larger particles. Smaller particles or smaller molecules are nothing but they are present in the filtrate. Okay. Let us, I hope all of you understood. Let us go to the next one. Are you all understanding? Okay. Let me proceed to the next one. I hope all of you understood up to here. We have discussed about so far about this thing, the biomolecules. Metabolites we have finished, primary metabolites which are compulsorily required for the survival of the cell or organism. Secondary metabolites, they are not required by the cell to survive, but they don't have any specific functions as such, but we are making use of it. Whatever the product it is, humans, we are making use of them. But for the living organisms, not exact functions we know. Then these biomolecules, the types we have got. Living tissue is grinded in trichloroacetic acid using mortar and pestle, and the slurry was strained. Whatever the liquid that is collected in the jar is called as slurry, and it is strained. Strained means filtered. Then we are getting the acid soluble pool. All of you can see here, acid soluble pool is there and acid insoluble pool is there. What is acid soluble pool? Filtrate. What is acid insoluble pool? Retentate. Those two you can understand here. Later they have given the molecular weights also they have given. Let me tell you the molecular weight later. But what you have to remember here, in the filtrate portion which substances are there, and in the retentate portion which substances are there. Already you know larger particles will be there and smaller particles are present in both of them. Filtrate, smaller particles, retentate, larger particles. Now let us see, I have written here 18 and 800 Daltons. In this I have written that molecular weight is 18 to 800 Daltons, and in this I have written molecular weight of 1000 Daltons. In order to understand that, first of all let me show you another slide saying that, see here basically we have categorized that molecules into 2 types based on the size. I told you size and solubility matters. Size, if it is small, that small sized ones, micro, is nothing but small, smaller molecules, where these are present in the filtrate. So micro means small molecules and they are present in the filtrate portion, and their examples are simple sugars, a normal sugar, then nucleotides, then amino acids, then fatty acids and glycerol. So these all will be there in the filtrate portion or acid soluble pool, and these are all called as micromolecules. You know why all these are called as micromolecules? Because of their small size. They are small size. And this smaller size micromolecules, basically they will be having a molecular weight of 18 to 800 Daltons. MWT is nothing but molecular weight. So all these compounds will be having 18 Daltons to 800 Daltons. Daltons is a specific unit which is used to measure the molecular weight of any molecules. So basically here, if the size of the molecule is small, it is called as micromolecule, and what should be the molecular weight? 18 to 800 Daltons. Okay, that is the maximum, this is the minimum value and this is the maximum value, 18 to 800 Daltons. So if any molecule is having a molecular weight in between these two values, then it is considered as micromolecule, and it will be there in the filtrate portion. No need for you to search anywhere. Obviously, it will be there in the filtrate portion because it has very small size. Next, coming to the macromolecules. Macro meaning is big. Big size. So the big sized molecules are called as macromolecules. Definitely all these ones, they will be having more than 1000 Daltons. More than 1000 Daltons is the molecular weight. So any substance, if it is having more than 1000 Daltons, okay, that molecules, any substances you can take which are having that molecules, and that molecules with more than 1000 Daltons are called as macromolecules. And generally this macromolecule should be there in the retentate portion. You know, they cannot be filtered, so they should be there in the retentate portion. You can find them generally in the retentate portion. But the problem comes with one kind of biomolecule, that is lipids. This last one. This is very unique. Okay, why I am telling you that this is very unique means, like lipids are actually micromolecules, but they will be present in the retentate portion. It's not basically a macromolecule, but because of solubility, this is the only one micromolecule present in retentate. This is important for the NEET point of view, because other than this, lipids, polysaccharides, nucleic acids and proteins, all of them, they will be present in the retentate portion, and they will be having more molecular weight, that is more than 1000 Daltons. But lipids, actually their molecular weight is just 18 to 800 Daltons, but yet they are present in the retentate portion. Do you know why? What is the reason behind that? Because of the solubility. They are not soluble in the acid. Which acid we used for that solubility? Trichloroacetic acid. So these are acid insoluble. As they are acid insoluble, they will not be there in the filtrate portion, but they are there in the retentate portion. Okay. So except lipids, except this, all others, these three are together called as macromolecules, and including that lipids here, these are called micromolecules. Why I have chosen like that means because of the solubility and portions. All of them are present in the retentate portion, and all these are present in the filtrate portion. Because of that solubility measuring, the solubility means that filtered, which are not filtered. So these are all filtered, they are present in the filtrate portion. These are all not filtered, so they are present in the retentate portion. That's why I have classified like this. Otherwise, these are all micromolecules along with lipids. This is not the macromolecule. This is the only one micromolecule in this list of retentate. Polysaccharides, nucleotides and proteins, all of them, they are the macromolecules. So this is how the biomolecules have been classified based on the molecular weight. Let us see the previous slide. Yeah, you can see now here clearly that biomicromolecules are there and macromolecules are there. In the micromolecules, we have the minerals, gases, salts and nucleotides. In the macromolecules, we have proteins, nucleic acids, polysaccharides, and you know that these lipids will come under the micromolecules. So, minerals means whatever the mineral ions are present, like chemical ions like H plus, Cl minus. In the chemistry, you must have learned that hydrogen ion, chlorine ion, okay, all those ions will come under that minerals. Then salts. Salt is nothing but NaCl, sodium chloride, like that different salts will be there, and gases like CO2, O2, nitrogen, all of them will be coming under the gases. Then nucleotides. These nucleotides I will be teaching you in the next this one after this slide only, just they are the building blocks of nucleic acids. We will be seeing that nucleotides. Then not only nucleotides, you have learnt already here amino acids. Then you have learned that lipids are also coming under this. I have kept the arrow, lipids are coming here, and even simple sugars are also coming here. These are all micromolecules, and proteins, nucleic acids, polysaccharides are the macromolecules. So now we have completed this session saying that the molecular weight of 18 to 800 Daltons is there for micromolecules, and for macromolecules the molecular weight is more than 1000 Daltons. These ones will be having the more than 1000 Daltons molecular weight. So have we completed all the things in this flow chart? I hope all of you understood. So coming to this one, the micromolecules and macromolecules. Okay, let me see. What about between 800 to 1000 Daltons? 800 to 1000 Daltons, the category is not there now. Just we have the categories up to 801 and above 800, but NCERT is saying that 1000 Daltons. Okay. Lipids is the only one in macromolecules who has no, it's not who has. Lipids are the ones which are present in retentate, but it is the micromolecule. I hope all of you understood. Who is that hell pirate? Sleeping and crying as well. Lipids are the only micromolecules that are present in the retentate portion. Understood all of you, ma? Yes? Shall I proceed to the next one? Give thumbs up, that is enough. Yeah, I'll be telling you that. We'll go to the next one. So metabolites are, we have completed, metabolites are there, and then the micromolecules and macromolecules which you have got by doing that filtration technique, filtrate and retentate. Okay. Now let us proceed to the next one. Yeah, all of you can see here. Very good, thank you. I can understand that you are understanding really good. Very good, great. So now coming to this, I have shown you in the before flow chart that minerals are also present, right? Minerals are present in the micromolecule portion. So that minerals basically are of these types. Elements, basically chemistry, okay? You might be thinking that botany teacher is teaching us chemistry, right? But you should know in this topic because it is related to biochemistry. This is a chapter in botany. So hydrogen, you must have seen that hydrogen already in the chemistry. Hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon. Don't ask me again that where are other minerals. We are giving comparison of these minerals only. In NCERT they have mentioned, so we are also going to see these minerals. Don't think that other minerals are absent, other minerals are also present. So all these minerals are present in the earth's crust also and in human body also. when we see Earth's crust, we have Earth, 3 layers are there. Okay, the outermost one is called as crust, next one mantle, next one core. So this is the outermost layer, this is the innermost layer of the Earth. So we have in the Earth 3 layers basically, from outer to inner. So the outermost one is the Earth's crust, where these minerals are present. So we have taken the Earth's crust one, okay, these minerals percentages. So if you see here, which is the highest ranking one, which is the most abundant one in the Earth's crust means, you can see here oxygen is having the highest percentage. 46.6 is there for oxygen. Okay, next second highest. So this is the rank first rank in the NEAT, but it is the first mineral in this section. In the Earth's crust, the first rank goes to oxygen with 46.6%. Second rank goes to, if you see here, silicon is having the second, 27.7. Second rank goes to the silicon in the Earth's crust. Okay. So now coming to this, you can see all other minerals as well, like the next category if you want to take, it will be the 3.61 calcium, and next one 2.8 sodium is there. Okay, after that you have 2.1 magnesium is there, and next one 0.14, that is hydrogen is there. Then these two are equal, like carbon and sulfur. So this kind of minerals are basically present on the Earth's crust. Crust is nothing but this outermost layer of the Earth. That is the percentages. They will be asking you in this area, like which is the most abundant mineral in the Earth's crust. Okay, which is the last one, negligible one, which is very little, that is nitrogen. So in the Earth's crust, first rank goes to oxygen, second rank goes to silicon, and the last rank, okay, is going to nitrogen. So these are the compositions of minerals in the Earth's crust layers. Now let us see our body, human body. In human body, which minerals are present? Yeah, correct. Oxygen is the highest and the least is nitrogen. Second highest is silicon. Generally they will be asking ascending order of these minerals or descending order of these minerals. So for that sake, you have to clearly practice this. You can make a list of this, the order of the first letters, like which is first and all. O, oxygen. Second highest, you know, OS, silicon, OS. Okay, like that you can prepare an ascending order or descending order so that you can remember. Okay, next that's what, aluminum. Don't think of other minerals because other minerals are also present, but that percentages are not given. Aluminum is also present, aluminum is present, so many other minerals are there which you will be learning in chemistry. In botany, these many minerals are enough because we are seeing just the Earth's crust part. Okay, now let us go to the human body. In human body, we have highest, just again for the NEAT perspective, these are important. So the highest in the human body is which one? 65%. That is again oxygen. So oxygen only is the first one even in the human body. You know, we will be respiring oxygen. The oxygen gas only will be respiring. Oxygen in human body, it is the highest, yes. Yes, in the Earth's crust, aluminum is also there, but we are not considering that part because it is not there in the human body. Here, this is a comparison between the Earth's crust and human body. In our human body, aluminum is not there because it is toxic. Okay, it cannot be there in the human body. That is the reason why we are not supposed to use the aluminum vessels also. We cannot cook or we cannot eat in the aluminum vessels. Aluminum is present in the Earth's crust but absent in human body. It is not there in the human body. So I am not going to give you that. Okay. So now the first rank goes to in the human body for oxygen. And can you tell me the second one which is there? Carbon. This is the second rank, second highest. Okay, third one, it goes to the nitrogen, third place. Okay, fourth place goes to the calcium. Okay, and 5th place goes to hydrogen, 6th place sulfur, 7th place sodium, and the last one magnesium. Okay, percentages wise. Then the last one in the human body, which is negligible, is the silicon. So they will be comparing those two things, like Earth's crust and human body, they will be comparing and they will be asking. I will ask you just one question, please answer to this. Okay, which is the second highest element, mineral present in the Earth's crust? The options are nitrogen, silicon, carbon, oxygen. Second highest in the Earth's crust. The comparison of only these minerals are mentioned in the NCERT, so these many minerals are enough. You have other minerals as well. Okay, silicon is the second highest, very good, correct. Name the mineral element which is first rank in both Earth's crust and human body. Name the mineral which is the highest in human body as well as Earth's crust. My second question, name the mineral which is highest in both Earth's crust as well as human body. Very good, oxygen. Very good. Keep rocking, yes. Good, correct. So oxygen is definitely highest. You can see here, this is highest. So just ascending order, descending order of these minerals only you have to learn. Okay, according to NCERT, these many minerals are important because they have given comparison of these two in the Earth's crust and human body. That much is enough. I hope all of you can understand that the elements which are present in the non-living and living matter. So in the living matter also these are present, and in the non-living things also these are present. Non-living things are also made up of these materials, generally the minerals which are present. So these minerals you have to learn ascending order, descending order, the comparison between the Earth's crust and human body as well. Okay, I hope all of you understood. If any doubts you can post, or else you can give a thumbs up. Shall I proceed to the next one? Before going to the next session, I would like to ask you one thing. All these ions, okay, they are inorganic ions or organic ones? Yeah, thank you. They are all organic or inorganic. Whatever this table, the ions are there. Will you call them as organic or inorganic? Which ones are these? Good, thank you. Yeah, which ones are these? Organic or inorganic? All this table contains hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium and silicon. These are all organic or inorganic? Okay, some of you are typing like organic. Okay, the mineral ions whichever I have listed here, if you take individual ions, they are all inorganic, but they can be involved in the formation of organic compounds. All these are inorganic. This is correct one. All these are inorganic, but they can be used in the formation of organic compounds. So when you get any ion, okay, no, all are inorganic. They can be used for making of organic compounds. Okay, but this table is giving inorganic mineral ions. This is inorganic chemistry. You are going to learn all about all of these in the inorganic chemistry, not in organic chemistry. In organic chemistry, you will be learning about the macromolecules, which are made up of CHO together, not single one. CHON, CHONP, CHONPS. So though all will be there together. So then it will become organic. If only one is there, that is inorganic only. Try to understand. These are all inorganic, but they can be used to form organic compounds. Understood? All are inorganic. Okay? Okay, now let us see how the organic compounds are formed. That's what, not carbon is organic, carbon C is an individual mineral element, it is mineral element which is an inorganic element, but that is used to synthesize organic compounds. All these are minerals before the table and they are all inorganic. They can be used to synthesize the organic ones. Okay. Carbon is not organic, carbon is also inorganic only, but it is used to synthesize the organic compounds. Try to understand that. Okay. In chemistry, you will be learning about all the details. Not only carbon, everything whatever I have written here, they can be involved in the formation of organic compounds. That table, all of them can be involved in the formation of organic things. But as individual elements, mineral elements, they will come under inorganic, individual elements. But if they are combined with any other element, then they result in the formation of organic ones, generally, which are present in the living organisms. Okay? They don't have that independent existence like that. My name is Geetha. Why do you need my name? Okay? Listen to the topic and try to understand the matter. Yeah, organic and inorganic materials. The organic ones are actually important ones which are formed by several molecules together, several inorganic molecules or mineral elements together. Inorganic ones, they are not having many minerals, only one or two will be there. That number will be varied. See, for example, water is inorganic. I'll tell you a best example which you already know, water. Water is inorganic. Inorganic, it is formed by just hydrogen and oxygen. Okay. So that is available in the nature freely, that is the inorganic one. CO2, again it is inorganic, carbon dioxide, gas available. Okay. Oxygen, O2, I am not talking about just oxygen O, I am talking about CO2, I am talking about O2. These are all inorganic. But later, by the combination of so many mineral elements in different composition, they will form the organic molecules. The details of organic and inorganic you will be learning clearly in chemistry. This is not pure chemistry. This is just a botany topic where you will be learning about these molecules functions. The details of further organic and inorganic you have to learn in the chemistry. You will be learning there. Okay? I hope you got my point, what I am trying to say. Let us proceed to the next one, biomacromolecules. I have told you that biomicromolecules are there and the biomacromolecules are there. Micromolecules means which are having the smaller size and their molecular weight is 18 to 800 daltons. Less molecular weight ones up to 800 daltons, they are called as micromolecules, and more than 1000 daltons, they are called as macromolecules. You can call, one of you have asked like 800 to 1000 daltons if they are there. They are intermediate molecules, but the classification you don't have at this level. That will be higher level. So you don't require that. So imagine according to NCERT, we have to follow NCERT. So according to NCERT, we have that molecules with a molecular weight of 18 to 800 daltons are called micromolecules, whereas above 1000 daltons they are called as macromolecules. Found in Earth's crust. Yeah, that's what? Compounds of carbon or the organic? Yes, correct, correct. The other details of organic and inorganic you will be learning in chemistry. Just try to understand, I have given a list of inorganic ions which are present in the Earth's crust and human body, that much is enough. And where they are present in the filtrate or retentate, that is important, where they are present. Minerals, gases, okay, all of them, salts, they all will be present along with amino acids, sugars and lipids. They are actually present in the filtrate except lipids. Okay? Okay, ma? So now let us see the next one. Micromolecules you already understood because they have the less molecular weight, 18 to 800. These are all macromolecules, so they will be having more molecular weight above 1000. Above 1000 they are called as macromolecules. So in this we have the names, proteins are there, nucleic acids are there, macromolecules, and then even carbohydrates are there. They are the polysaccharides, and then you have the lipids. But this lipids, as I told you, it is the micromolecule. For the sake of convenience we are writing like this because these lipids are present in the retentate portion. Retentate means acid soluble or acid insoluble. Retentate, acid soluble or acid insoluble. Lipids are acid soluble or acid insoluble. Yeah, acid insoluble. Very good, Amma. Great. Lipids are acid insoluble. Very good. Correct. That is the reason why they are present in the retentate portion, though they are the micromolecules. So this is the reason why I have kept the lipids under this classification, because they are present in the retentate. Okay, but they are micromolecules. Remember, this is very important. Lipids are micromolecules. So before going to that macromolecules, we should understand first of all, okay, the micromolecules. Let us see the first micromolecules here. Before going to the macromolecule, you should see the micromolecules, because any macromolecule, it should be formed by the combination of micromolecules. Macro means big one. So if you combine two small things, micromolecules, obviously you are going to get a big one, that is a macromolecule. So here amino acids are micromolecules because they have the small size and their molecular weight is 18 to 800 Daltons. 18 to 800 Daltons. So they are the micromolecules. These are present in which area? Filtrate portion. Or you can call it as acid soluble pool also. Okay. So micromolecules which are having actually smaller size, that 18 to 800 Daltons, they will be fused to form the macromolecules. We will see the macromolecule after fusion which will be formed later. But here try to understand that amino acids, here the amino acids heading is there. So they are actually the acid soluble ones, they are present in the filtrate ones. Lipids are insoluble in the trichloroacetic acid, because I told you the filtration technique, how you can separate the molecules. Okay. See, if I give you a leaf, if I ask you to burn the leaf, a dry leaf, okay, when you burn, whatever the ash you will get, that ash consists of inorganic substances. Ash, okay. All organic compounds will evaporate. So that's where we can actually separate organic compounds from inorganic ones. When I go to the lipids, I will be telling you, but first listen to me. The micromolecules, they are amino acids. So here micromolecules filtrate acid soluble pool and they have a less molecular weight, 18 to 800 Daltons. One example, amino acids. Amino acids means amino. The meaning of amino means nothing but amino group is present. This is the amino group. NH2 is called as amino group. You can see here NH2 is there, amino group. And this amino group will be always there on the left hand side. Left hand side. Okay. And this is the central carbon, H is there, and down variable group is there. Variable group means which can be changed. Okay. Variable group means which can be changed. Now, COOH means carboxylic acid group. COOH. This is the functional group for the carboxylic acids. In chemistry, you will be learning like acidic group is there, COOH, aldehyde group is there, CHO, okay. Then ketone group is there, C double bond O, like that you will be having in chemistry so many functional groups. So in that functional group, COOH is the one which is called as carboxylic acid group, okay. So amino acid, let us see the meaning. Amino means NH2 is present, this is present, okay. Acid means carboxylic acid group is present. So a micromolecule which is formed by the amino group and carboxylic acid group is called as amino acid. You can see the words amino and acid. So this compound is having one NH2 group on the left hand side and carboxylic acid group, that is COOH, on the right hand side. So if any organic molecule, if it is having one amino group and one carboxylic acid group, that is COOH, it is called as an amino acid. But these two are linked to what? The central carbon. The central carbon. Okay, let me show you. Here, the C is there. Carbon. Okay. Someone have given the message saying that okay, organic compounds will be having the carbon. That is true, because any of the biomolecules should be formed generally by the central carbon atom. Carbon will be there. So carbon valency, in chemistry you will be learning that carbon has a valency of 4 sides. 4 is the number, valency of carbon. So all the 4 sides, some or the other ion will be attached. That will fulfill the valency point. In the chemistry you will learn that. So here amino acids means, as I told you, amino means which group? NH2. NH2 is nothing but amino. So we have to add on the left hand side amino group. Right hand side, acid means carboxylic acid, COOH. This is a simplified version of amino acid structure. NH2 on the left hand side, right hand side COOH. And above hydrogen and down R. R is nothing but a variable group which you can write anything. These variable groups only will be deciding which amino acids are there. Very good, carbon valency is 4. So now if you write on the left hand side amino group and right hand side COOH group, then down one R variable group, it is called as amino acid. This R variable group, if you keep on changing, you will be getting different types of amino acids. Literally, we have 20 amino acids which are available naturally. 20 naturally occurring amino acids are present, which we need to synthesize proteins. So naturally occurring amino acids. Okay, how many are there? Naturally occurring amino acids in all living organisms? 20 are there. Okay, so the 20 will be different from each other in terms of variable groups mainly. At least one amino group, one carboxylic acid group should be there. More than that also can be there. Minimum 1 and 1. One amino group, one carboxylic acid group. Remaining, this variable group can be anything. Okay, in that way 20 naturally occurring amino acids are present in nature, which are definitely required for the formation of a protein. See, all our body functions, you are growing in size because of the protein intake. We should have the pulses, because the pulses or proteins are meant for body building. So that proteins are actually made up of amino acids. So amino acids are actually called as micromolecules and monomers as well. You have understood already micromolecules based on the size. Now I am going to give you one more term here, monomers. a single unit. Okay, that monomers are nothing but amino acids, and these are building blocks of, if you link the amino acids in the chain form 1, 2, 3, 4, 5 amino acids chain, this will be forming protein. Okay, so 20 type of naturally occurring amino acids if you link in different chain form, like not necessary that always first should be this one, second should be this one, but so many amino acids chain, if you link them together, it will form a protein. So protein monomers or micromolecules with which it is made up of, or building blocks of proteins are which ones? Amino acids. Building blocks of proteins, with what the protein is made up of? Amino acids. So protein is considered as macromolecule, or you can call it as polymer, because macromolecules are already having the more molecular weight and they are present in the retentate portion. So they are formed by the combination of amino acids, which are building blocks of proteins, or amino acids, and monomers, they are called as single units, are called monomers. So here we are linking so many amino acids. No, for example, I have linked 5 amino acids here. So many, you can attach 100 also or 1000 also. So the chain of amino acids will be called as protein, which is a polymer or a macromolecule. So after this, can I conclude saying that monomers or micromolecules examples are amino acids. Okay, or you can call them as building blocks also. For what? Monomers means the chain of monomers will be forming polymers. Okay, micromolecules chain, if they assemble together, they form macromolecules. Okay, amino acids, if you link them together, they result in the formation of protein. Okay, so with the help of that building blocks or micromolecules, okay, will be forming the macromolecules, or you can call them as polymers. So amino acids are the monomers or the building blocks of proteins. Proteins are macromolecules or polymers which are made up of amino acids. Have all understood this more? Amino acids are micromolecules and they are monomers, and proteins are the macromolecules and polymers. Okay? Okay. Let me show you the structure of different amino acids. Yeah, you can see here, very good. So amino acid means, first of all, you should have one amino group and one carboxylic acid group. That is the basic rule. And do you know the molecular formula of methane? Have you heard of methane? Anyone? Methane. Yeah, very good, CH4. So amino acids are called as substituted methanes. Substituted methanes. So methane is nothing but CH4, where you will have like this. So here you can see the first amino acid is glycine, which has just hydrogen. I told you R is a variable group. So if you remove R and if you place H, it is going to become glycine. If you remove the H, here in the glycine or R group, if you place it methyl group CH3, it will be called as alanine. If you place in the R group hydroxymethyl CH2OH, it will become the serine. So these three structures all of you should know because they are all given in the NCERT. Different example, three examples: glycine, alanine and serine. Glycine is formed by placing in the R group H. Alanine is formed by removing R and placing methyl group, and serine is formed by the hydroxymethyl placing in the R group. The structures are important. All the three structures you have to know. These three amino acids are important. Okay? The simplest amino acid is glycine. Now all of you can see essential amino acids are there and non-essential amino acids are there. Essential amino acids means our body cannot synthesize, so we have to eat them in the food. Cannot be synthesized by our body. These cannot be synthesized by the body, so we have to eat them through our diet. Example: lysine, leucine, isoleucine, tryptophan, etc. We have essential amino acids are 10. Some of the examples only I have given here. So essential amino acids, you know, both of them together 20. So 10 are essential and 10 are non-essential. Essential means our body cannot synthesize, so we have to eat them through the diet. Non-essential, our body can synthesize these. So no need to have the amino acids here. Just by eating our food, our body, inside our body, these amino acids can be synthesized. So these are called non-essential amino acids and these are called essential amino acids. So which ones we have to supply through our diet? Essential or non-essential? Essential should be supplied with the diet. Okay, so through the diet we have to have the essential amino acids. Okay, ma? So essential amino acids should be supplied through our diet only because our body cannot synthesize these amino acids. Non-essential can be synthesized by our body. Some examples: glycine, alanine, serine, arginine. You have learnt already glycine structure, serine structure and alanine structure. Glycine has H in the R group, alanine has methyl group in the R group, serine has hydroxymethyl group in the R group, variable group. So they are the non-essential 10 will be there and essential are 10 are there. Now let us see here. In the amino acids, you know, left side amino group is there and right side COOH group is there. So in different solutions, acidic solutions and basic solutions, if you keep the amino acids, okay, essential are 10, non-essential are 10. If you keep any amino acid in the acidic solution or basic solution, you will be getting the ionic form. The basic ionic form what you will be getting will be the Zwitter ionic form. When both the sides, see here, NH3 plus is given, okay, NH3 plus and COO minus. Just remove the H from this area. See here I am writing the H group, NH2, you know, here R group, here I am writing the H and here I am writing the COOH. Just take out this H and add it here. Then the compound what you will be getting here formed will be NH3 plus, okay, C, H, R and COO minus. This kind of ion which is formed by removing that hydrogen from the carboxylic acid group to the amino group, this ion is having both positive ion and negative ion here. This type of ionic form of amino acids is called as, this is called as Zwitter ion. Zwitter ion. Zwitter ion means nothing but in amino acid we have to have one positive ion area, amino group NH3 plus and COO minus, that is the Zwitter ion. Okay. So this is the zwitterionic form of amino acid. I hope all of you understood. NH3 plus and COO minus. Now that amino acids, if they are linked up in the chain form, it results in the protein I told you. So that these all dots are amino acids. Whatever the rings I have shown you here, all of them are amino acids. This is the chain of amino acids. If it is like a linear chain or a pearl ball chain, any ball chain if it is looking, it is a primary protein structure. Okay. If a chain of amino acids is there simply like this, it is the primary structure of protein. If that amino acids are linked like this, one here, two here, 3, 4, 5, 6. In different areas, if they are linking like this, okay, if they are linked in this form, this is called as secondary structure. It is not a linear chain. This chain will be showing extra bond. Here the bond present is peptide bond between the amino acids. That will form the primary structure. Chain of amino acids will form protein. It is especially primary and the bond is peptide bond. Primary means it looks like this and this bond is called as the peptide bond. In the secondary, this is the peptide bond only, but they will have even hydrogen bonds also. Secondary structure will have peptide bond plus hydrogen bond. So it will be showing some kind of foldings, okay, something like alpha helix. It will be looking like this. This is alpha helix structure. This is called beta pleated sheet. It will be linking like this, one after the other. The chain will be linked like this. So primary will have only peptide bond between the amino acids. Secondary will have peptide bond plus hydrogen bonds in the structure. That is secondary structure. Alpha helix structure and beta pleated sheet. This is tertiary structure. Primary, secondary, and this is tertiary. In this, some extra bonds will be there, not only peptide and hydrogen bonds. This time extra bonds will be there, like ionic bond plus peptide bond, hydrogen bond plus ionic bond, covalent bonds, van der Waals forces, so many extra bonds will be there. Hydrophobic bond, disulfide bonds, so many n number of bonds, whatever the bonds you know, you can write there. So if you have so many bonds in the structure, it will show different foldings. That is a tertiary structure. It looks like some kind of woolen ball. With all these bonds, that structure will be folded in such a way that it looks like a hollow woolen ball. Then you have even quaternary structure also. Two will be like alpha chains will be there like this, and two will be there like beta pleated structures. Two will be like beta pleated forms. So if you have each of them like one cube, it will form the quaternary structure. So we have basically four structures: primary, secondary, tertiary and quaternary. In primary, we have only peptide bonds. In secondary, peptide hydrogen bonds are there. In tertiary, so many extra bonds are there, but only one polypeptide chain, one chain only will be there. In quaternary, many chains are there with all the bonds. I hope all of you understood. Yes, ma? So next one, lipids. Lipids are micromolecules, but they are present in the retentate portion. Micromolecules, but present in the retentate portion. So lipids are classified into simple lipids, compound lipids and derived lipids. What is a lipid best? First of all, let us see that. Lipids are trihydroxypropane, glycerol. One best example of lipid is the trihydroxypropane. Okay. Primary structure and secondary structure, just I will show you once. You can see here. Primary structure, just chain. secondary structure alpha helix and beta pleated sheet, tertiary woolen ball, one chain only, quaternary 4 chains, 4 different bonds and 4 structures. Okay. Now let us go to the next one, lipids. In the lipids, the best example of lipid is glycerol. It is called trihydroxypropane. See the name tri, tri means 3, hydroxy means OH, propane means which will be having 3 carbons, straight chain. So here first carbon, second carbon, third carbon, 3 OH groups are present here, and carbon valency you know it is 4, CH4. So CH2OH, CHOH and CH2OH together it is called as glycerol molecule. It is a simple lipid. Okay. Its name is trihydroxypropane. Tri means 3, hydroxyl group means OH, propane means 3 carbon chain is present. Next one, all of you can see here, the glycerol is linked to one fatty acid, then it will form monoglyceride. Glycerol, one glycerol molecule linked to one fatty acid. This is the fatty acid palmitic acid, which has 16 carbons in its structure including COOH. 16 carbons are present in the structure. One glycerol plus one fatty acid is called monoglyceride. Mono means how many? 1. So one fatty acid only is present. If you add for one glycerol 2 fatty acids, it will become diglyceride. Can you tell me in the same way triglyceride means? 3 fatty acids are added to the one glycerol. 3 fatty acids are added to one glycerol. Okay. One glycerol plus one fatty acid is monoglyceride. One glycerol plus 2 fatty acids is diglyceride. One glycerol plus 3 fatty acids is triglyceride. And the bond which is present between these two, this is called ester bond, which is formed by removing one water molecule. You know glycerol structure already, CH2OH, CHOH, CH2OH. Just remove that hydrogen from one and OH from one, it will become water, water will be removed, and this fatty acid which is given is palmitic acid. You can write like R1 COO. Here they have written R1 COO, R2 COO. Here R1 COO, R2 COO, R3 COO. Monoglyceride, diglyceride and triglyceride. So just number of fatty acids will go on increasing. Glycerol will remain only one. One fatty acid means monoglyceride, 2 fatty acids means diglyceride, 3 fatty acids means triglyceride. And what is a bond that is present? Ester bond. This is compound lipids. This one is called as phospholipid because it has the phosphate. In the same structure, triglyceride which is there, this is the first fatty acid location, this is second fatty acid location. In the third fatty acid location, they have the phosphate, so it is called phospholipid. Example name is lecithin. This is called as lecithin. Very important, by looking at this structure, you should identify that it is lecithin. It has CH2 and CH3 thrice. This is a phospholipid which is generally present in the cell membrane. You know, each cell will be surrounded by the plasma membrane. So that plasma membrane will be having lecithin, which is a compound lipid, and it is an example of phospholipids. Fatty acid, glycerol and phosphate. They are made up of 3 things. So this lecithin is an example for compound lipid which is present in the plasma membrane of the cells. I hope all of you understood this. Examples ma. Next, one more example of lipid is cholesterol. You know, so many people will be having cholesterol in their blood below the skin, okay, inside the blood vessels. This is the cholesterol, which is one more a lipid like compound, or we can say derived lipid. By looking at this structure, you have to make out this is cholesterol. Just 1, 2, 3 hexose rings will be there, which are having like this 6 corners, but this has 5 corner one. Okay, and this has a branched chain attached to it like this. So cholesterol, by looking at this structure you have to identify that it is the cholesterol. Next one, nucleotides. You have got already the nucleotides which are building blocks in the micromolecules. This term was there, nucleotides. Nucleotides will form the DNA. DNA is a nucleic acid. So nucleotides are the monomers. DNA is the polymer. Okay. So monomers or nucleotides, the polymers which are formed by that is the DNA. So nucleotides will come under micromolecules and DNA will come under macromolecule. The chain of nucleotides will form the DNA, deoxyribonucleic acid. So in this deoxyribonucleic acid, what are the contents that are present? Nucleotides or heterocyclic compounds. Heterocyclic compounds means, hetero means different, cycles are present. Hetero means different cyclic compounds. So many cycles are there. If you see here, so many colored rings are there. So if they are made up of different type of cycles in their structure. So heterocyclic compounds. And nucleic acids, 2 types are there, deoxyribonucleic acid, RNA ribonucleic acid. Both are made up of nucleotides only. So nucleotides are the building blocks of nucleic acids like DNA and RNA. So far you have learnt amino acids are the building blocks of proteins. Now nucleotides are building blocks of the nucleic acids like DNA and RNA. Okay. Amino acids will be forming proteins and nucleotides will be forming the nucleotides. Lipids are micromolecules only, they are formed by fatty acids and glycerol. Different types of lipids you have learnt. Now let us see here. In this, one particular nitrogen base is made up of, here we have 3 things. If you can see, nucleotide is made up of 3 things: sugar, nitrogen base and phosphate. Okay, 3 things will be forming the nucleotide, which is again micromolecule. 3 things will be there. Okay. Nucleotide will be made up of sugar, nitrogen base and phosphate together. See here, nitrogen bases are given as purines and pyrimidines. They will be having this type of structure. Adenine, guanine. They have 2 rings. Okay, heterocyclic compounds. Then thymine, cytosine, uracil, these are having just 1 ring compounds. These are called pyrimidines, these 2 are called purines. So you have to remember these things. Nitrogen bases are purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. Cytosine, thymine and uracil. Coming to the sugars, sugars which are present are deoxyribose sugar and ribose sugar. Okay, phosphate is nothing but that PO4 will be added. This is the phosphate. This is the sugar, ribose sugar and deoxyribose sugar, and this is the phosphate, and the purines adenine and guanine. With the first letters you can write Ag, thymine, cytosine and uracil. The combination of all these will form the nucleotide. So nucleotide is nothing but 1 sugar, 1 nitrogen base and 1 phosphate. In nitrogen bases you have purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. I hope all of you understood. The names are given. Adenosine. This is adenine combined to sugar. So this is actually called as nucleoside. If you attach for this the phosphate, it will become adenyllic acid, which is a nucleotide. These are important here, nucleotide and nucleoside. Nucleoside is formed by the combination of one sugar and one nitrogen base. Nucleotide is formed by the combination of sugar, nitrogen base and phosphate. The names are given, these structures are given in the NCERT. Just once again will be teaching you in detail biomolecules, so don't worry. The things which I have told you, you have to remember. This is how the DNA will be formed with the bonds which are formed. Okay, I have told you already, protein is made up of a chain of amino acids with peptide bond. In the lipids, fatty acid and glycerol will be linked by ester bond. Okay, in the lipids will have ester bond, and proteins will be having peptide bonds. Then the sugars, the carbohydrates which are there, polysaccharides, they have the glycosidic bond, sugar, one sugar plus another sugar, glycosidic bond. Simple sugars are also micromolecules. Then hydrogen bonds are present between the nucleic acids, particularly nucleotide chain between nitrogen bases. These are hydrogen bonds, but these ones are again different bonds. Between the sugar and phosphate, this is sugar and phosphate, it is ester bond. Between the sugar and the nitrogen base, this is the glycosidic bond. Okay. In sugars, only glycosidic bond is there. Sugars will be linked, like glucose will be linked to form starch, cellulose, etc. That bond is glycosidic bond. Amino acids are linked to form proteins by peptide bond. Lipids are formed by the combination of fatty acid and glycerol by the ester bond. And polysaccharides, nothing but that sugars are formed by the combination of simple sugars, glycosidic bond. These are the nature of bonds which are present in different compounds. The next one is enzymes. Enzymes are generally basically proteins only. Most of them are proteins except one or two. So these are just proteins, equal to proteins, almost equal to proteins. Most of the proteins, okay, will be forming, they will be functioning as enzymes. So we can say most of the enzymes are proteins. And each enzyme, if a conjugate enzyme or holoenzyme is there, it is formed by protein part and non-protein part. That non-protein part is called cofactor, protein part is called apoenzyme. Cofactors are of two types, organic and inorganic. Organic ones and inorganic ones. In organic you have coenzyme and prosthetic group. Coenzyme is easily separated, very easily, loosely attached. Prosthetic group is firmly attached. And inorganic ions, they can be attached in however they want, they can be attached. This is how they will be attached, apoenzyme, cofactor. Those two will be assembled together to form the holoenzyme, a complete enzyme. Apoenzyme is the protein part. Okay, cofactor is the non-protein part. This is the protein part, this is the non-protein part. That means this is made up of amino acids. This is non-amino acid portion. Only this is the amino acid one, this is the non-amino acid one. Both of them will combine to form the enzyme. And the best part is, if you remove this cofactor, enzyme will lose its activity. Enzyme loses its activity if the cofactor is removed. And enzymes will be functioning like this. They will be converting the reactants or substrates into products. In chemistry, we will be calling substrate as reactants. In biology, botany, the reactants are called as substrates. okay, and the products are formed by the enzymes. This is how the enzyme will be acting. This is without enzyme, the reaction graph, it will be like this. With enzyme, it will be like this. The more things about these you will be learning later. In order to understand that, you have to learn so many other things as well. This is the essence of today's class. I started with biomolecules. We went for the amino acids which are forming the proteins. Then you have learned nucleic acids are also macromolecules or polymers. They are made up of, there are two types, DNA and RNA. DNA, we have different types, left-handed, right-handed, RNA, rRNA, mRNA, tRNA. Next we have in the proteins primary structure, secondary structure, tertiary and quaternary structure. Then I have told you about lipids, which are the micromolecules present in the retentate portion. Simple lipids, conjugate lipids and derived lipids I have shown you. Then saturated fats are there and unsaturated fats are there, okay. Conjugated lipids are also there, like phospholipids, lecithin. These two things already I have told you, phospholipids and lecithin. Then derived lipids, I have shown you cholesterol structure over there, okay, steroids, cholesterol structure. Then in polysaccharides, polysaccharides are nothing but sugars, carbohydrates, basically these are carbohydrates which are made up of simple sugars with a bond, glycosidic bond between them, and that sugars can be composed of same monomers, homopolymer, or different sugars. If they are made up of same sugar, then it is homopolymer. If they are made up of different sugars, it is heteropolymer. Then lastly we have learned that enzymes, most of the enzymes are proteins only, and they will be made up of two components, apoenzyme and cofactor. So both of them in combination together, they will be forming the enzyme. So if you have any doubts, you can ask me now. Just you have learned the overall brief explanation of biomolecules. Just you can go through what is the structure of amino acid, okay, what are lipids, the basic things, which are monomers, which are polymers, which are micromolecules, which are macromolecules. Those things if you learn, that is good, where they are present in the filtrate or retentate and all. Later we will be explaining all the other necessary details. The basics are enough. Biomolecules will be learning in detail after coming here, and it is you will not be learning like in this span of time only. We will be taking like more than 10 hours or 15 hours to complete the chapter, because it is lengthy in NCERT. So just a brief glance only I can give, so which I gave you, the brief one. So just go through the structures which are given in the NCERT. I hope all of you understood. Thank you so much.